This is Dr. Alam, Assistant Professor of Biochemistry and Biotechnology, North South University. So today we will study lecture 5, and so today we will study details about the action of enzyme. So in the previous classes we studied a little bit about the enzyme. So what is enzyme? Enzymes are macromolecular biological catalysts that are basically proteins and that accelerate or catalyze chemical reactions, and the molecules at the beginning of the process upon which enzyme may act are called substrate, and the enzyme convert this into different molecules called product. So enzymes are classified according to the type of reaction they are catalyzed, and all the enzymes have formal EC numbers, enzyme commissions numbers and names, and most have trivial names. Okay, so international classification of enzyme belongs to 6 classes. So oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligases. So oxidoreductase, this catalyze the transfer of electrons, hydrogen or hydrogen atom, and involved in oxidation reactions. And transferase, they transfer group or group transfer reactions, and basically they transfer functional groups. Hydrolase, they catalyze the hydrolysis reactions, that means transfer of functional groups to water, transfer of groups to basically hydroxyl group. And lyases, those are catalyze the lysis reactions across a double bond, and they add groups double bond or formation of double bond by removal of groups, and isomerase, they catalyze the transfer of group within molecules to isomeric forms, and basically they involve in molecular rearrangement. And ligases, so they catalyze the formation of carbon-carbon, carbon-sulfur, carbon-oxygen, carbon-hydrogen bonds by condensation reactions coupled to ATP cleavage, and they basically catalyze the joining of two substrate molecules. So enzyme specificity, this refers to the tendency of enzymes to catalyze a specific type of chemical reactions, and some enzymes are absolutely specific and only catalyze one chemical reactions. So based on that, there are two models of enzymatic function. One is lock and key model, another is induced fit model. So in lock and key model, so there the enzyme fits exactly to one substrate and complementary shape like a lock and key. To repeat, in lock and key model, each enzyme fits exactly on one substrate and they have a complementary shape like a lock and key. And another model is induced fit model, and this model is the extension of lock and key model, where some enzyme can bound multiple substrate, and interaction between enzyme and substrate induce a change to fit the substrate. So here enzyme can change their active site to bind exactly on the the substrate. So what are cofactors? Cofactors are substances other than the substrate whose presence is essential for activity of an enzyme. So small molecules that bind with the enzymes and are necessary for enzymatic functions are called cofactor. There are two types, inorganic or organic. So inorganics are metal ions, and organics are called coenzyme, mostly the vitamin. So here you can see this is the inactive protein where cofactor binds and produce a binding site or active site for the substrate, and then substrate can bind with it. So with that cofactor, these types of reaction will not be started. So some enzymes do not have any additional component to show full activity. However, other enzymes require non-protein molecules are called cofactor to bound for activity. Cofactors can be either inorganic or organic. So inorganic molecules are called metal cofactors, and organic molecules are two types called coenzyme or prosthetic group. And inorganic compounds are two types called metalloenzyme and metal activated. So what are the organic cofactors? Organic cofactors be either prosthetic group or which are tightly bound to an enzyme, or coenzyme which are released from enzyme's active site during the reaction. That is, they are loosely bound with enzyme. So examples of coenzyme include NADH, NADPH, and ATPs. These molecules transfer chemical groups between enzymes. So an example of an enzyme that contains a cofactor is carbonic anhydrase with a zinc cofactor bound as a part of its active site. These tightly bound molecules are usually found in the active site and are involved in catalyze. So here it catalyzes the reaction carbon dioxide plus water to produce carbonic acid, and enzyme protein plus enzyme protein plus cofactor. This is called holoenzyme. So there are two important terms, holoenzyme and apoenzyme. If enzyme bound with cofactor, this is called holoenzyme. If there is no cofactor, just only one enzyme is present, this is called apoenzyme or apoprotein. So in details about the models of enzyme action. So as we studied previously, so enzymes have long been studied for a biochemist, and there are two models for how enzyme action occurs, like lock and key models and induced fit models. So this is very important, and the important part of this model is that the substrate fits exactly into enzyme's active site like a key into a lock. And in induced fit model, the substrate does not exactly fit into the exact active site of the enzyme. Instead, to accommodate the substrate, the enzyme changes the shape of its active site so that the substrate can fit into the active site of the enzyme. And these types of enzymatic action requires the enzyme to use energy in order to change its shape. So this is an energy consuming process. So here the mechanism of the actions. So the lock and key hypothesis, it fits between the substrate and the active site of the enzyme is exact, and like a key fits into a lock very precisely, and the key is analogous to the enzyme and the substrate analogous to the lock, and temporary structure called enzyme substrate complex is formed, and product that have a different shape from the substrate, and once formed they are released into the active site from the active site. Okay, so temporarily when substrate bind with the enzyme, they produce enzyme substrate complex. This is a different product, and that have a different shape from the substrate, and when product is produced, they are released from the active site and leaving the enzyme free to become attached with another substrate. So this explains enzyme specificity, and this explains the loss of activity when enzyme denatured. So about induced fit model, some proteins can change their shape. Look at here, the active site of the enzyme and the substrate side, the binding site of the substrate is not same. So here, according to the substrate, when a substrate combines with an enzyme, the enzyme induces a change in the enzyme's conformation, and the active site is then moduled into a precise conformation with the substrate and making a chemical environment suitable for the reaction, and then enzyme substrate complex is formed, and then the bonds of the substrate are stretched to make the reaction easier, and finally enzyme product complex is formed and the product is released, and enzyme will regain its previous structure. So this explains the enzyme that can react with a range of substrate of similar types. So enzymatic reactions are very rapid. Look at here. So the hydrogen peroxide breaks down to water and oxygen. It occurs 600,000 times per second with catalyst. To achieve maximum product per unit time, need enough substrate to fill the active site. So optimal temperature, pH, and enough enzyme increase the rate of reaction. So what are the factors that affect the enzymatic activity? Like their substrate concentration, temperature, optimal pH, enzyme concentration, and also inhibitors. So enzymatic activity increase with the substrate concentration and more collision between substrate molecules and the enzyme. Enzymes activity increase with the temperature. Warmer temperature causes more effective collision between enzyme and substrate. However, hot temperature destroys the enzyme or denature the enzymatic enzyme conformation. Most enzymes are optimized at a particular pH, so extreme pH also denature the protein or enzyme. Enzyme concentration. The activity of an enzyme increase as the concentration of the enzyme increase if you continuously supply the substrate. So look at here, what is the action of pH and enzymatic activity. So how pH influence the enzymatic activity. So rate of almost all the enzyme catalyst reaction depends on the pH, and most enzyme exhibit optimal activity at pH value between 5 to 9, and higher or lower pH value than optimum value will cause ionization of the enzyme, which result in denaturation of the enzyme. So we can say each enzyme has an optimal pH that maintains its normal configuration. A changes in pH alters ionization of the site change, eventually resulting in denaturation. So how substrate concentration influence the enzymatic activity? If you plot the rate of reaction with the substrate concentration, you can see if you increase the concentration of the substrate, the rate of the reaction will increase until all the enzymes are saturated with the substrate. So this is the point of saturation, and increased concentration does not affect the reaction rate. So faster reaction, but it reaches a saturation point when all the enzyme molecules are occupied. So how temperature influence the enzymatic activity? Enzymes are most active and optimal temperature, usually 37-degree temperature in humans, and shows little activity at lower temperature, and loss activity at temperature above 50 degrees centigrade, as denaturation occurs with loss of catalytic activity. So as temperature rises, enzyme activity increase because there are more molecular collision. Enzyme activity declines rapidly when enzyme is denatured at a certain temperature, resulting in a change of shape of the enzyme. So that's the end of the lecture 5. See you in the lecture 6.